Thank you, Anshul, and thanks for the organizers for inviting me here and having the opportunity to present you some of our work. We are placed in Munich. In Munich, there's an Institute of Human Genetics, and in front of the seminar room of the Institute of Human Genetics, since about 20 years, we have huge panels that are 1 meter wide and 10 meters tall. And when you approach those panels, you see this, and this is a part of the X chromosome as it was sequenced. And since 20 years, we're still, unfortunately, many times clueless as to which variations in our 3 billion nucleic acid long genome can cause disease. And this is important for understanding predispositions to common diseases, to understand which variants are causing also rare disorders, and of course as well, which variations can drive cancer. What you can do today, thanks to large biobanks, you can do one thing that is perhaps the most simple thing to do, is to compare the frequency of variants here on the x-axis against their effect on traits. And if you take a trait that has any relevance to health, such as cholesterol, you typically see a trend like this. So on the right-hand side, the common variants can associate, some common variants associate with health-relevant traits, but often with a very small effect. And this is because of selection. If a variant will have a strong effect, then it will be selected against or fixated if it was relevant for health. On the other extreme, you do find sometimes rare variants that have effects, and there one can see very strong effects. There is still a corner here, which a part of the data that seems to be missing, and this part is due to statistical reasons. So that if a variant is rare and have a very minor effect, then it's hard from a statistical point of view to associate it with the trait. When we look at the type of variants that we see among this in this plot here, you have on the common variants and the small effects a lot of non-coding variants, and mapping them to genes and pathways is difficult. On the left-hand side, on the other hand, you're more often finding variants that affect directly protein sequences, sometimes even loss-of-functions variants. So finding the genes and the pathways is more easy. At the same time, also the effects are stronger. So this gives good entry points for developing therapies. And here I'm showing here ApoB, and this is a gene which, when it's loss of function, convert individuals carrying those loss of function lower cholesterol. And that's one of the targets investigated by pharma to lower cholesterol. Rare variants also show up in rare disorders. So rare diseases are diseases that affect less than 1 in 2,000 individuals, but there's a large number of those, 5,000 to 8,000 distinct rare diseases, so that collectively they affect a large fraction of the population. Often, not always, but often these disorders have a genetic cause, and it's absolutely essential for these affected families to pinpoint what is the genetic cause of the rare disorder. Once this is found, this will enable the genetic testing, so the families can know who in the family is carrier of a potential pathogenic variant. Couples that are at risk can sometimes then perform pre-implantation genetic screening. The families can be connected, the clinicians following those families also, they know they work on the same disease. And because you have then the root cause of the disorder, there is a clear rationale for their developing therapies. However, as of today, there's still, for the majority of individuals that undergo whole genome sequencing and are suspected to be affected by a Mendelian disorder, the causal variant is not identified. So to summarize very simply, the major trend I mentioned to you Variants that have a high impact on health due to selection are rare in the population. What I didn't show you is that the converse is not true. Most variants that we see are neutral. The vast majority of the variants are rare. And so identifying which of these rare variants have a high impact requires more than the mere statistics. And this is where we need you guys. We need machine learning, and we need further information to be able to go beyond just the frequency observations. And so I will show you today three type of data that we can use to prioritize variants that can be pathogenic. And these three aspects of research we perform in our lab cover, on the one hand, how we can leverage genomes of multiple species. I'll tell you about dependencies analysis from DNA language models. On the other hand, we'll look into expression, in particular aberrant expression. And then finally, how we can use biobanks and connect rare variants together with phenotypes. So first, I'll talk about these DNA language models and their dependencies. There's a very strong motivation in the field to leverage, even better than we have done so far, genomes that are coming from biodiversity genome sequencing projects. We can see now tens of trillions of bases that are sequenced across eukaryotes and prokaryotes. And these are fantastic data to perform self-supervised learning, so learning that is done on these, what we call unlabeled data from a machine learning point of view. That is, we have the input, the X, for our phenotypes, the genomes. We don't necessarily have all the phenotypes on all the species, but we can train representations of these data that perhaps are helpful for performing then supervised learning tasks. There is a fundamental reason why this has high potential, and that's because the genomes that we see from the species today are the product of evolution. It means there is constant mutation occurring on those genomes and constant selections, so that the elements that are functional, that are important for fitness, are not randomly distributed in these genomes. They are positively or negatively selected, depending on the context in which they occur. So this is a particularly attractive type of data for self-supervised learning from these non-uniform distributions to extract features that will then be helpful for our supervised learning tasks. It's an old idea, of course. This is the idea of comparative genomics, but we can revisit comparative genomics today with deep learning techniques. And so many groups, including ours, have been embarking into this topic. I'm showing you now just a few slides on the paper we published last year, and I presented also 2 years ago at ISMB. And that's an algorithm that we call species LM. And this is a simple transformer which takes as input DNA sequence, and it's trained from a masked nucleotide modeling task. So we mask individual nucleotides in the input, and the model is tasked to give a probability for each of these 4 nucleotides at the positions that are masked. It is called species LM because we also provide the token for the species, and this allows us to then train on genomes that are very evolutionary very far. Thanks to the species context, the model can adapt the predictions to the species it sees, and so we can use genomes that are so far that's part of their regulatory code has evolved. His work was done by Alexander Carolus, Johanna Singel, and Denise Gankin. I won't show you much, but I'll show you one picture that you see a bit what happens when you train such a model. We are looking here at how certain the model is about every base in a particular section of the yeast genome. What you see here is a section that is starting 5' of the start codon of a ribosome gene, RPL36A. It goes into the promoter of the gene, and then it goes into another gene that is tandem to it. So you're reaching the 3' untranslated region of this gene. The model didn't have those annotations. It only has DNA. But what he finds is that it's pretty sure of itself about the identity of certain bases that happens to be transcription factor binding sites here for this promoter. And in the UTR of the gene 5' to it, we find motifs that are known for RNA binding proteins that recognize elements in the RNA. So the model figures out the context of these sequences and provide more confidence as to elements that are actually functional. The papers go in way more details about this. We find distinctions from the model between transcription factor binding sites that are bound or not bound. We find differences between species. And so this indicates that these models are indeed learning interesting representations. And we and others then use those models as foundation models to perform later on supervised learning tasks and revisit tasks such as predicting splicing, gene expression, pathogenicity, etc. And so we have ended up with performances that in several instances were outperforming state-of-the-art models here in yeast, and others have also looked at the same approaches for different downstream tasks. But we felt this is not just should not just look at this from a foundational model point of view. It seems that these models have learned a lot of biology that perhaps we should be probing a bit better than just by performing prediction tasks. So we broadly ask ourselves, what sequence context do these DNA language models capture? Because they're good at predicting the identity of a sequence given a context. And so here, Pedro, Thomas da Silva, and Alexander Carolus embarked on a project where they looked at this very simple idea here, that is asking how much does a query nucleotide affect the reconstruction of a target nucleotide at another position according to the model. And so I'll show you how this, so first of all, I want to emphasize that this is very model agnostic. We are not looking into the parameters, into attention matrices or so. The only thing we require is that the model gives us this conditional distribution. So this is something we can apply very generally. And therefore, we also applied it for many foundation models, not only our species NM, also models from others. So first, I want to show you actually the model that the locus, which for us was an eye opener, and that's a tRNA, so a transfer RNA. You see here on the left-hand side that the models, when exposed to that sequence, is pretty confident about the identity of the bases. And we know that this tRNA is forming a structure, and one of the primary, or the secondary structure is shown here on the right-hand side. And so that position here, the query, is known to bind in a Watson-Crick fashion with this target, which is about 80 nucleotide further down. When we ask the model, what if this query base were not a T, but an A, it changes probability for the other base from A to T, and vice versa, and C to G, etc. So the entire Watson-Crick role is actually learned. The model has not learned by heart that sequence. It has really learned the base pairing. So what we decided then to do is to take those matrices and extract and aggregate statistics, and we went for the maximum absolute log-odd ratios. So then what we can do is we can traverse an entire locus, taking every base as a query and every base as a target, and record these maximum log-odd ratios. And as you can see on this tRNA here, we get, in a completely unsupervised fashion, the entire contact for all the arms of the tRNA. We are not only getting this secondary structure, actually the tRNA is a 3D thing. It folds into this L-shape molecule, and the L-shape is important for the tRNA to fit into the ribosome, and this is thanks to other tertiary contacts, which are here where my pointer is. And it turns out that with our species LM, one of these tertiary contacts also shows up in the dependency maps. And when we use other models that are focusing on RNAs, we even can get all the 3 tertiary contacts that then completely solve the structure. So these concepts are old. Maybe you know about covariation analysis. You can focus on the tRNA, align it, and look across evolution at covariations. But what is very important to see here is that this entire analysis was not focusing on tRNA loci. We've just looked at non-coding regions in yeast, in fungi. We never performed sequence alignment. We didn't tell the model, look here and align the sequences. And we didn't enter the biophysical rules. So everything is just learned directly from the dependencies. So we then went on and computed maps across the entire genomes and looked at what we see. And we saw patterns. And I invite you to look at the paper to see the variety of biology that is recovered. I'll just highlight a few today. One is that we often find blocks on the diagonal of those dependency maps. And this is well illustrated with this promoter here in yeast, promoter of the gene SMT3. First, I show you the reconstruction of the DNA language model. And as you can see, you have a poly-A stretch that is pretty well reconstructed with confidence, and then a few motifs which we could label thanks to what we know from yeast. If you look at it from a dependency point of view, the picture is a bit different in the sense that the poly-A stretch has these very diffuse dependencies. And this is because if you change a single A to another base in a stretch of 20 A's, the model will not really change its probability for the other A's. Whereas if you are, in contrast, in a transcription factor binding site like CBF1, changing the identity of one nucleotide will have a strong effect on the identity of the others. So as a consequence, you see that there's while all those sequences are a bit undistinguishable from just the reconstruction, once you go to dependencies, you have a different pattern. And we decided then to exploit this pattern to define convolution filters that we even didn't train. We just go with a square matrix along the diagonal and record the top quartile of diagonal elements. And just doing this in yeast, you find you can call TF binding sites at the same level of accuracy than if you were to go to a database of position weight matrices that have been identified using experiments, using ChIP-seq experiments, and scan those promoters with the position weight matrices. Those are not the most fancy models, but they are models that had needed experiments to be built, whereas the scan of the diagonal here is independent of any experimental data. So this is quite remarkable, and it beats the reconstruction, which leads way more other bases that are not relevant for the TF binding prediction task. So that suggests that we could look into regulatory code of other species that are not studied as well as yeast. On the RNA structures, we found that these anti-diagonal patterns, which you've seen in the tRNAs, are characteristics for RNAs that have these stems. And you can see clear enrichments for RNAs that are annotated as structural. We collaborated with Danny Incarnato, who can probe in cells, and he did that in E. coli, whether an RNA is folded or not, at which positions it's folded. And that's an assay that is called DMS. And this assay basically induces mutations in positions that are not paired. So we had basically four predictions in E. coli that could be validated with this assay. And this includes things that are not so straightforward to predict with classical bioinformatics tools, like this pseudoknot here. Another application is really for us in machine learning. We all, after building, or many of us, after building foundation models, which hope will be helpful for our downstream tasks. And often you realize that it was helpful, perhaps, once you've done the downstream task. And it's pretty hard to know which of the decision in the foundation model or after is actually the one that makes the difference in your final model. So with these dependencies, we can probe a foundation model for the statistical dependencies that it captures, and we can test whether these capture the type of biology perhaps that we are interested in. So here, if you're interested in tRNAs and you look at different models, so RINALMO is a model that is trained on a database of RNAs and gets pretty good recovery of tRNA secondary structures, the species LM I mentioned you. And then there are other models that performs a bit not as good. For example, the nucleotide transformer is binning the genome into 6 mirrors that are not overlapping. And that induces very strong dependencies between the bases within each of these blocks. The most important take-home of this benchmark was that the models that are trained on only the human genomes are pretty poor at capturing tRNAs. whereas those that have multi-species are much better. And there's a good control here because nucleotide transformer comes in with the same parameter size with a multi-species or without multi-species. So back to our problem of variant influence and how we can use that to prioritize variants in, for example, in genomes of patients. The observation we made for this poly-A stretch, in the promoter, where we see a difference between having A, and it's easy to guess that the next base is an A, and having a base which, when it's mutated, change the probability of the others, like in the transcription factor binding site. This contrast we thought could be perhaps be interesting to leverage. And why? Because most of the approaches that use foundation models to prioritize variants come from the idea that something that is unlikely is bad. But if you have many, many A's and one is not an A, maybe it's not as bad as if you have a transcription factor binding site and one of the letters is not the right one. So being easy to guess doesn't mean it's important, but having a huge influence on the reconstruction of the other bases may be more important. And therefore, we devised the variant influence score, which is the average influence that we defined before in the window around a particular base. So we look at this first on ClinVar. ClinVar is a database of variants that are associated with diseases, and you can contrast the pathogenic variants and the benign variants, non-coding here. And you see a clear separation using our influence score. Importantly, we get a better separation if we use the influence score than if we use the likelihood ratio of the base. So this is done with using the very same DNA language model. We don't change the DNA language model, we just use the influence score instead of a reconstruction. You see, however, that we are not outperforming alignment-based conservation at this prioritization here. Perhaps one reason for this is that all pathogenic variants we have in clinical databases are based from analysis that themselves used directly or indirectly conservation. So there's perhaps a bit of circularity there. And to have an analysis that is independent of this issue, we looked at a saturation mutagenesis. The model we're using here, called species LM metazoa, is trained close to the start codon of metazoan genes. So it contains typically 5' UTRs, also promoter regions. So we looked at the promoters of this screen. What you see on the left-hand side is that qualitatively, the reconstruction probability seems to agree with the importance you have in this reporter assay in which every base in this piece of DNA has been mutated, and the effect on transcription on a reporter gene is measured. However, the reconstruction itself does not outperform ranking of these variants by alignment-based conservation. If instead, and it's again using the very same DNA language model, we use the dependency scores now, we outperform the conservation and make a much better ranking than using the reconstruction. Finally, we looked at a supervised model, a state-of-the-art supervised model called Borzoi. So this is a model that is trained on ENCODE, on a lot of labeled data, as we have in human and mouse. And when you run it to score the variance of the saturation mutagenesis screen, you get a score that is not significantly better than the influence score. And we could also show that we can combine both in a very simple linear combination for each promoter and get a bit of an improvement, suggesting that perhaps these DNA language models have captured elements that the supervised models Borzoi didn't have. More work is to be done on this, but this is interesting to see already. Okay, so the implications of this first part of what I'm talking about today are several-fold. So first, we believe that this approach is interesting to look at regulatory code, especially in species for which we do not have experimental data, or in layers of regulation, such as post-transcriptional regulation, perhaps, where the space is less probed than in transcription. There are also very strong evidences that these models learn interesting features for RNA structural biology, and a lot of work is done in RNA structural biology using foundation model nowadays. So you think that attacking problems such as pseudonodes, alternative structures, or tertiary structure perhaps will be more accessible. These principles allow us to probe foundation models without fine-tuning, and that's an interesting feature. And then we came up with, or we observed that the influence of a nucleotide on reconstructing others, rather than their own reconstructability, seems to correlate better with fitness, and that's also perhaps a concept that is interesting to use in the future. And finally, it gives us a way of visualizing sequences, and simply making observations is sometimes the great starting point for doing discoveries. So the dependency maps as a way to look at sequences can be also an interesting tool. Okay, so move now to the second part of my talk, which is about aberrant expression prediction. So the field is making a lot of progress in interpreting variants, including non-coding variants, but still, as I mentioned earlier, in the majority of the cases for individuals that receive a genome sequencing from a suspected Mendelian disorder, we are not able to pinpoint the genetic cause. And so alternative ways are needed. And I've established a long-term collaboration with the lab of Holger Prokisch since several years now, where we are not only looking at the genomes or exomes of those patients, but we also perform omics data on them, including RNA-seq and proteomics. And from a modeling point of view, the simple idea that we have followed is the following. Bluntly said, in rare disease diagnostics, the outlier is the signal. When you look at a data set from rare disease, often you don't have a very large cohort of individuals that suffer from the same mutations or the same gene, even though perhaps sometimes you see similar phenotypes. So you have rather data that looks like on the right-hand side, an individual may have for a particular gene a level of expression that is aberrantly low or high, on a splicing event, rather than enough data to do a case control. So instead of working with differential expression tools, we need outlier detection tools, and that's what my lab has been doing for several omics layers. And the last one we have, and we have a poster in this conference about it, is called PartRider. So Daniela is presenting this. And that's a work together with Ines Scheller. And so the idea that we have followed along the years is follow the following paradigm. You take data, you model this with an autoencoder that gives you a reconstruction of your data, and then you contrast the observation to the reconstruction. And the difference is these residuals are then modeled with some distributions, and we look into the tail probabilities, and we identify there the events, the extreme events. Our latest tool we use for proteomics not only model the intensities that you observe in data, but also the missingness. As you may know, in proteomics, missingness does not occur at random. So there's some information about intensities, but non-trivial. And the beauty of these autoencoders is a very flexible way to deal with that. Daniela, thanks for having that idea and having pushed for it. And so I'll show you this is indeed helpful. And the way we benchmark PartRider, but also all the other outlier detector we worked on, is by using genetic variants as orthogonal data. So the autoencoder always works on a purely omics data set, in this case, purely proteomics, never been exposed to the DNA of the individuals. And we use annotations of variants which we know are likely to cause an outlier. They're never for sure causing an outlier, but there should be an enrichment for those. So for protein, it would be missense variants that are likely affecting protein stability, nonsense-mediated decay, splicing, which often result in nonsense-mediated decay. And then we can rank by different methods what is the most likely to be an outlier down to the least likely. And we can see the enrichment, the proportion of outlier that have one of those variants suspected to disrupt protein abundance. And as you can see, the PartRider, which models missingness, does perform better than the one that does not model missingness, and alternative methods. And here, one of the general trends, it's always very helpful to model this latent space to correct for the covariations, including biological covariations that you see across individuals, to really restrict to what is unique to your sample. This concept, as I said, we applied it for not only for proteomics, also for RNA expression, for splicing. We had the chance to collaborate with many consortia on rare disease, and so by now we have shown that this helps increasing the diagnostic rates depending on how well the affected or the accessible tissues capture the biology of the disease by +5 to 15%. Given that this disease plateau a bit at 50%, this is a substantial contribution. And 3 members of the lab are now have now set up a spin-off called Omics Discoveries, where they plan to sell those services. Okay. So what I showed you now is that we are able to establish tools to detect outliers, and we make use of the genome to benchmark those. That's kind of orthogonal data. And now we can pivot this. What we will do is we will tell this as a ground. We take the outliers as a ground truth. They're not perfect ground truth, but they're good proxies. And we're going to build algorithms which take the genome as input and predict outliers. And we've done this on splicing, a tool called AB Splice, and our latest one, done by Flo Salvima, look at aberrant expression, RNA expression, and it's called ABEXP. And what we've done is we applied our outlier detector on the entire GTEx. So we have called outliers for over 11,000 samples, and that gives about 11,000 underexpressed outliers. And then we take every gene and we filter for rare variants, and we see whether we can predict which of the genes and in which tissue will give an outlier. We can do this even in a continuous fashion. You don't have to purely binarize. The benchmark is done on the binarized fashion, but the model is built in a regression fashion. The first thing we observe is what other people also observe. The group of Steve Montgomery at GTEx have made those observations. One of the major signals for outliers that we see in the genomes are not transcriptional, they're post-transcriptional. It's nonsense-mediated decay, which is the occurrence of a premature termination codon that leads to RNA degradation. Now, all other ways to get to NMD, in particular around splicing, often also leads to NMD. And then there are other things that I'm not showing here, but I'll show you later, such as structural variants that are also important. So these are features that we've been using then in ABEXP. We have looked into transcription. We've used Informer. And the signal that we could find is the signal that is confined very close to the transcription start site. It's called the core promoter. And this is where single variants, because often variants are SNVs, single variants can lead to an outlier. Variants that are further away, we don't find them enriched. And this signal is, let's say, consistent with promoter AI, the recent paper from Illumina, will also find that the enrichment is close to the TSS. Another aspect that was important, this is also an idea that Daniel MacArthur published some time ago. We see that variants will lead to nonsense-mediated decay if and only if the actual part, the exon where it occurs, is used in the tissue. And so we leveraged a map of isoforms that is provided by GTEx to refine our predictions to be isoform specific in a condition specific, in a tissue specific fashion. And that improves further the predictions. The last part that was perhaps surprising was that we see a large variation in terms of gene expression variability, even after correcting as much as we can with our autoencoder between genes. Well, that's well known. So this gene here, LTB3, this is a gene that is a gene that you can see that is very tightly regulated. There's very little variations between individuals. This olfactory gene, when measured in blood, perhaps not surprisingly, varies a lot. And as a consequence, if you have a fold change of expression here, you're not likely to make an outlier. Here, a little 30% of change, and you're already outside the physiological range. So one needs to take this into account to predict aberrant expression. And so altogether, ABEXP is a model that integrates these features, also aberrant splicing, also structural variants, and then increase the precision over using traditional tools. The papers do more. So we do more in the paper. We we also show that this gives a good ranking of pathogenic variants in ClinVar. We show that we can integrate RNA when it's accessible from an accessible tissue to predict in a non-accessible tissue, so the RNA together with the genome. And finally, we also show that this is helpful to detect genes that are associated with common traits in biobank data. And to explain you this, I'll move to the last part of my talk. That is how we can use genomes together with human real-world phenotypes from biobanks to improve our way of modeling how variants affect fitness and predisposed to diseases. So back to this plot with which I opened my talk today. As I mentioned, we have this relationship between variant frequency and effect size. And we have here on the left-hand side something that is very interesting from a biological but also a therapeutical point of view. We find genes that are associated with the traits. But this is just at a single variant level, and so we may have more power if we're able to pull information across multiple variants within the same gene. That's a concept called burden test that I'll explain in a minute. But the motivation for that is to identify these effector genes, so places where we see really a mechanistic relationship with the trait. And it's also interesting to be more sensitive in this area to perform better polygenic risk scores, to be better at identifying individuals which are at high risk for a trait. So the concept of a burden test is a pretty simple concept. It exists in many variations, but the basic idea is the following. So you consider a gene, and then you annotate variants that you see in individuals. Those are all your individuals. And you provide a score to the variant. So the most simple thing you could do is just ask, is a variant likely to lead to loss of function of the gene? Yes, no, and count how many you see. But you can also have more weighting of those annotations. And then once you have obtained such a score of the gene, you can associate the burden together with the phenotype, either in a directional way or with a variance testing to ask whether some genes tend to be more mutated or less mutated than others along with the variations in the phenotype. So we revisited this. This is a collaboration with Oliver Stegle at the DKFZ and Eva Holtkamp, who is a PhD student in my lab, and we collaborated with Brian Clark, who is now an independent team leader in the Institute of Oliver. And we decided to use deep learning to have a more data-driven way to train the way we would compute those burden scores. So these gene impairment scores that we give for every individual as a function of variance. And the idea we follow is a very simplistic idea. We will tell that there is a universal function that just takes variant annotation that doesn't know about the trait, that doesn't know necessarily about the gene function, or will score at the gene level a degree of impairment. And once this is done, this is the part where the deep learning will act. On the right-hand side there, we do simple linear models, adjusting for covariates, population structures, and so on. But the right-hand side here is once you have an impairment score, you just do a linear combination of those gene impairment scores to predict your trait. Nevertheless, this is a powerful approach. It's simplistic, but it allows us to really train using a lot of data these gene impairment scores. For this, we use an architecture called a set neural network. The
Should be.
Okay, so I'll restart everything. Okay. Yes, and so, so that is what you see. Now, when we train our models, deep that we have a continuous score here, which has several advantages. As you can see, first, we are able to split this putative loss of function variance into some that happen to actually indeed associate with low LDL and others that were higher. When we looked at it, we saw that the CAT score was different for those two types. So the model has learned how to integrate this. And the beauty of this is that the model has learned this not only looking at this gene and this trait, but a set of about 300 gene trait pairs. So it's a very general function, which we can then apply to other tools. It's not just overfitting on this. And so here to other situations. So then if I show you now a pair, GLPD1 and alkaline phosphatase, that was not used during the training, we also see this continuous score that associate with the trait. So this is important. That shows that we have a generalization of the gene impairment score across traits. We benchmark our model against state-of-the-art models that also use multiple variant annotations to perform our rare variant association testing. And these tools are not using the same logic. They're based on variance testing. We found that we were on quantitative traits. We were also calibrated, but we were a bit more sensitive, and we could recapitulate better our predictions using a larger data set that was not used for the training. That's on the quantitative traits. Where we really saw a difference is when we moved to binary traits, especially impaired, imbalanced, such as disease traits in UK Biobank. These tools that were existing were not really not calibrated in those cases. So they report tons of genes, even for disease that have very low prevalence, which is extremely unlikely. You can appreciate on the QQ plot that probably the p-values are not calibrated here. Whereas we can plug our impairment score as an allele dosage in a tool called Regeni, which is a tool that is very robust for these cases. It uses first logistic regression. It has good ways of dealing with relatedness and population structure, which was not accessible for those tools. So as a conclusion, our tool here is a tool that allows us to do in a more robust fashion rare variant association testing for disease traits, and we think that this can be also helpful for this type of research. So in conclusion, with DeepHarvard, we have established a procedure for rare variant association testing that learn how to integrate variant annotations. We found that the approach has a good calibration, even for imbalanced binary traits. And then a few points that I think can go beyond doing those analyses. Having learned on UK Biobank, that is on the data set where we have genomes and measurement, we can learn an impairment score which is free of human annotations. And that makes a difference. I work a lot on rare disease. And what we often have is things like ClinVar, and this is built by people that have used old tools, manual annotations, to populate those databases. So we keep learning the old tools. They're very circular. Here we have a gene impairment score, which seems to work broadly for a large set of traits. And so I'm looking forward to see how it will be helpful for rare disease diagnostics. So far, we only train on exomes. We are now working on genomes. And another limitation, perhaps, we are not using information at the GIL level. In a sense, it's a powerful approach to be agnostic of gene function, but we can go beyond. And so if you're interested in that, you can go to Anna Starovoit poster, D545, where she will show a method whose preprint is here that allows us to go beyond and integrate a gene function. With this, I'd like to thank the team I work with. I'm very lucky to work with smart and motivated people, and so that's really a pleasure. Some are here. I hope you will meet them. I'll thank, of course, our funding, and I thank you for your attention. I'll finish with this slide, so you have a few take-homes and pointers, and I'm ready to answer questions.
All right, great. I think we can move to questions. We have about 12 minutes for questions. Start with, do we see anybody here? I guess you can walk up to the mics. Go ahead, number 2. Mic number 2.
Thank you for the nice talk. Want to ask about these genomic language models, especially for dependencies between nucleotides that are species-specific or cell-type specific. Do these models have weaknesses there because they are trained on hundreds of species? And if a dependency is only occurring in one specific species in one cell type, how do they capture those effects?
So for there's no cell type logic in a DNA language model because it's the blueprint of the genome is the blueprint of all cell types. The cell type interpretation may make sense if, let's say, you find an interaction with something that you can identify from other reasons as being the transcription factor binding site of something that only occurs in one cell type. One could interpret this way, but technically it's just a statistical dependency that comes from looking at many genomes, and you don't really have anywhere a cell type label. You do have, however, the species label, and we have not looked at, well, in a sense, we have looked at that before having figured out the dependencies, so it won't show up like this, but if you look at our first genome biology paper, there we do show that some transcription factor binding sites are reconstructed in some species, but not others. And we know that it's not a perfect correlation, but it does correlate with the fact that the species where it's not reconstructed are those that do not have the transcription factor, and vice versa. And so in that sense, yes, we do have dependencies that are species-specific.
Does MSA also recapitulate the dependencies?
The MSA, so there's GP and MSA, which, okay, glad to ask. You ask, there's a few slides on that. The MSA, so multiple sequence alignment-based large language models, or at least GP and MSA, is predicting the identity of a base given an alignment. And we've explored with dependency what happens there. If you look, for example, at the tRNA, you get not at all the dependency, even though the reconstructions are very good. And I think the reason is that to predict that base, the model tends to look up at the column, and it's not really needed to learn an entire representations of motif and dependencies. The alignment has already done the job. And so it's not so good, and it's a bit our conclusion at the moment. It's perhaps not very good at representation learning, while it may be a better way to do conservation because this leverage the fact that these alignments capture ortholog pieces of DNA, and so selection occurring at one base may be better represented like this. So we'll see in the future what happens, but my feeling is that the more you use the alignment, the better you capture orthology, and you make good at fitness score, for example. Whereas if you're interested in representation learning, you probably want to go a bit more away from alignment.
Thank you.
You're welcome.
All right, mic number 1. Go ahead. Yes.
Yeah, I have a question. On this dependency detection that you showed, how far can the two nucleotides be in a genome
Yes.
still be detected?
So we are limited by the size that those models take as input. We have worked in the lab with things that went up to 2 kb. There are models that are larger. So that's one aspect. And then the other aspect is, so beyond what they can technically capture is what they actually manage to capture. And so we've done a bit of studies of how these things decay as a function of distance. There's a clear power law. And we you get very little interactions after a few 10 bases. But we do have things along the entire range. So the full 1 or 2 kb that can be captured. We have, for example, examples of donor site and acceptor sites of introns that are captured. Beyond, not yet.
So if you take it, let's say a TF in a bacterial genome, could you identify the multiple sites that bind that TF?
You don't get interactions. We didn't find interactions between the binding sites of the same TF. A little bit of crosstalks between some of the transcription factor binding sites could be observed, but we don't have very systematic analysis of this.
Thank you.
Welcome.
All right, number 2 again, right there.
That's very impressive work.
Thank you.
I understand that the dependency score is different from, for example, the attention metrics. Have you looked at like how closely they are associated, and what is the advantage of this dependency score over, for example, attention score?
We have not looked too much into the attention. The problem with the attention is that you have attention at all layers, so you need some ways to aggregate this, and it's also extremely bound to the type of algorithms you use. But others have done that. What we have explored different ways. So our definition only need this conditional distribution, so it's agnostic of the implementation. That's a good thing. The bad thing, it requires all these in silico mutagenesis, so it's a bit costly. What seems to give a good proxy is to look at the embedding of the last layer, but not just at the position of the base you're interested in. That correlates well with the log ratio, but at the entire region. So when you introduce a SNP, do you affect the entire representation? That is a good proxy of our score, and actually it ranks very similarly. And perhaps it captures the same mathematical underlying theme, perhaps.
Yeah, thank you.
You're welcome.
All right, number 1, right there.
So you were showing in one slide for variant scoring, you were combining dependency scores with Borzoi predictions, and the dependency scores were from which model? Was it from nucleotide transformer?
Sorry? The dependency score that I was showing there it's one from us. It's a species LM that we train on metazoans. Okay. And it's focusing on the region that is 2 kb 5' of the start codons.
So my question is that what is stopping us from combining the training of Borzoi-like models? Like when we train them, doing the loss calculation, if we do the language modeling as well. Can you comment on that?
I guess there will be many papers of the community on this soon. I don't know. Nothing. I think perhaps we were a bit hesitant to go so far, and now there's way more evidences that this has potential, and I assume this will come very soon.
Yeah, thank you. And second question is that, so your lab, when you train the DNA language models, you don't train on the whole genome, but specific portions
yes.
that have more functional impact. But the trend in the field is that, okay, we train on the whole genome, like most of the DNA language models. So what do you think, that has impact on the nucleotide dependencies when we gradually increase the training data and start training on the whole genome?
I would also like to go whole genome. When we see people that are doing whole genome, they do a sampling that is not very uniform, so that they also start close to genes or inside genes. I think it's basically a way to provide during training first more informative sequences. But ideally, hopefully, we'll get a big model that have seen everything, and that hopefully also will give us more long-range dependencies.
Yeah.
Maybe I'm dreaming.
Number 2.
Bonjour, Julien. Thank you for the nice presentation. I was wondering, so in the beginning, you showed that these outlier detection methods are better to compare, basically, to find extreme cases, extreme examples compared to case control. And then you showed the gene impairment scores as a quantitative score, where then again you return basically to the binary case control or regression setting. Do you think there might be an opportunity to use the gene impairment scores in these outlier detection methods, especially for low data regimes?
To use what? Sorry.
to use the gene impairment scores, since they're a quantitative value again, so you could see them kind of as another omics, and to use them instead of one of the other omics layers you use in Outrider, Podwriter.
Perhaps I didn't think this way. But what I think it is maybe a value of applying these autoencoder approaches on phenotypes, and that perhaps will clean up a lot of correlations that we see, for example, in blood biomarker measurements. And maybe they will correlate better with the genes, but we only started to explore that.
Thank you.
Okay, number one.
Okay, thanks. So I have a question about the last part of your talk on burden test on genes. And you mentioned that in future work, you're going to look at the genomics. So I wonder how you apply your method on non-coding regions. What would be the similarities and the differences compared to the method that you showed on genes?
We'll probably focus on the gene body plus minus 5 kb, because these are regions that are relatively obviously associated to a gene, even though it is known that some enhancers can talk to other genes, right? So this is not too trivial, but we will first start with these basic assumptions and see where this brings us.
Any more? Okay, number 3, last question.
Hello, can you hear me? Thank you for the very nice talk. I have one simple question. You mentioned that the use of multiple species genome improve your prediction results, right? So, but in the phenotypes, have you considered using any multiple species phenotypes, not only for the exome phenotypes?
I would love to. I think it's a very exciting direction. We don't have so good data for species, but perhaps in cattle also that there's something
there
that
is
could
mouse genome and fish, and this across multiple species ontologies. You will find a lot of information out there.
Yeah, there's another, okay. But not between individuals, between species. Okay, we could talk after, perhaps.
Sure,
Thank you.
Okay, let's thank Julien again.
Thank you.